Hello, good afternoon. Hope everyone is having a good Saturday. So today, Zach and I will talk to you about how do flu vaccine cause immunity in people and why it doesn't always work. I'm Wei Ping Cao.
And I'm Zachary Endy, and thanks for joining us today. So before understanding how flu vaccines work, you may wonder why we care at all about the flu enough to make or take a vaccine in the first place. So the first is seasonal illness. Every flu season, millions of people get sick, seek medical attention. And these are numbers from this past year's flu season, which were relatively low, but you still had hundreds of thousands of people in the hospital and tens of thousands of deaths, most in older adults, but about 1%, a little bit less than 1% were children in the U.S. So it's pretty serious every year. In addition, you also have pandemic potential, which unfortunately all of us understand what a pandemic means nowadays. This is pictures from 1918. It looks like it could have been from yesterday. So in 1918 was probably the biggest pandemic. It was 675,000 U.S. deaths approximately from influenza virus infection. But relatively smaller pandemics have also occurred more recently in 1957, 1968, and 2009 with the swine flu. In addition to pandemic potential, there's also deadly strains like bird flu or avian flu, avian influenza. These are all the same things, which have really high mortality rates, but typically don't cause pandemics worldwide. There are effective antiviral drugs for influenza. Here I'm showing some that are recommended for this year's season by the CDC, like Tamiflu, but they're not a cure. They need to be taken to work best within a day or two of the onset of symptoms, and drug-resistant strains can also occur, making the drugs less or not effective at all. So I hope you're somewhat convinced that the virus is worth looking out for, though as much as you try, you won't actually see it because it is quite small. So to put the size in perspective, I'm going to zoom in to compare it to other small things. This is from the University of Utah's website. So we're going to compare it to cells and bacteria and see how small it is. Look for the picture in the upper right here. This will eventually come onto your screen as you see what size it is. So it's smaller than a grain of salt, single cell organisms, skin cells, chromosomes, bacteria. There it is. I'm going to show you that again just so you can see how small that is. It's tiny. Basically, the size of the virus to us is roughly the size of us to the Earth, to give a little bit of a perspective. But just because it's so small, it doesn't mean it's not powerful and complex, and Wei Ping will fill you in on some of the details.
Zach, you're right. We definitely cannot see the virus by eyes. Here are some key details of the influenza virus as discovered by scientists. The picture shows the structure of influenza virus, and they are made of a viral envelope containing the glycoproteins wrapped around a central core. So these glycoproteins are hemagglutinin, we call it HA, and neuraminidase, we call it NA. So HA is the protein that mediates binding of the virus to target cells and enter into the cell. And NA is involved in release of the virus particle from infected cells by cutting the sugars that bind the mature viral particles. So the central core contains a segmented viral RNA genome and other viral proteins that package and protect this RNA. So there are 4 types of influenza virus, influenza virus A, B, C, and D. So type A influenza virus are divided into subtypes based on the type of HA and NA. So there are 18 known types of HA and 11 known types of NA. Virus subtype is described by letter and number. So H3 and 2 here means type 3 of HA and type 2 of NA. Other names we might be familiar are H1N1, H5N1, and H7N9. So how do we name different viruses? We know there's a lot of different strains of virus. So the human influenza virus is expressed in this order. You have A here shows in the figure, that's virus type, and where the virus was isolated is a Fujian here, and number 411 here is a strain, a lineage number. And the year of a virus was isolated is 2002, and followed by virus subtype H3 and 2. So to protect from influenza virus infection, vaccination is the most successful and cost-effective public health tool. Here are some benefits of the flu vaccination that you should be aware of. So the flu vaccine prevents thousands of flu-related hospitalization every year. And it also can reduce children's risk of ICU admission by a significant percentage. The flu vaccination is an important preventive tool for people with chronic health conditions, and it also reduces a pregnant woman's risk of being hospitalized with the flu by average of 40%. And also, flu vaccine reduces the severity of illness in people who get vaccinated but still get sick. Therefore, hope convince you that to prevent and control of seasonal influenza, we should go and get vaccinated. And CDC's Advisory Committee on Immunization Practice, ACIP, recommended that all persons aged older than or equal to 6 months who do not have contradictions should get routine annual influenza vaccination. Emphasis should be placed on vaccination of high-risk groups and their contacts and caregivers. So a licensed vaccine appropriate for age and health status should be used. And as we know that there are many different kinds of flu vaccine products available on the market. And Zach will tell you more about that.
Thanks, Wei Ping. So the flu vaccines begin with a virus chosen by the World Health Organization, which is predicted to circulate for the coming year. And there are a number of ways you can take this strain and make a vaccine product. The earliest ways that this was done is you grow the virus in the lab and you inactivate it chemically. Some people call it kill. So you kill the virus so it can't infect you, and then you inject it into someone's arm, and that's called a whole inactivated virus. Another way is to attenuate the virus or weaken the virus so the virus is too weak to cause illness, and that's the flu mist, which was FDA approved in 2003 that gets sprayed up your nose. You can also take the whole inactivated virus and then break it up with a detergent into its component parts before injecting the vaccine. Fluzone falls into this category. It's a very common category of flu vaccine. FDA approved in 1980, but as you can see, others were approved later on. You can also go one step further and purify specific viral proteins like the HA or the hemagglutinin, which gives the H1N1 or H3N2, the hemagglutinin. So that's a virus surface protein. And FluBlock was approved in 2013. That's an example of a vaccine like that. And these are the main products out there, but besides these, which have been FDA approved, there's also other methods that you could use to produce vaccines. So you could also make them with DNA, RNA, with pieces of viral proteins smaller than these, or you can make virus-like particles, which are missing the genome so they can't replicate. I'm not going to go into it, but the RNA, this is the vaccines that are being discussed right now with COVID. There's an RNA-based vaccine that seems to show efficacy. So these are the products which happen at the end of a long process. And the process, as I said, starts with this chosen strain, and then it gets grown in chicken eggs, which is the traditional method, or it gets grown in a dog kidney cell line. Yes, that is a Cocker Spaniel, not in the dog itself, just in the cells that originally came from a kidney from a Cocker Spaniel. Or it can be grown in insect cells. So I hope this isn't too much bad information for you. Everything gets purified out. So in the Fluzone is an example that gets grown in the eggs. The FluCellVax gets grown in cells. So that's why it has the cell in it, FluCellVax. And then in insect cells through genetic modification, that's how FluBlock is made. So the whole process takes about 5 to 7 months. So that's why you have to basically choose a strain in about February. And you end up with about 200 million doses in the US. So that's as much as we're going to say about how they're made. And now that you know that, Wei Ping will explain a little bit of what happens once you get injected.
Thank you, Zach. So when you get a flu shot, the vaccine injected into the muscle cells induce alarm signals. And those are cytokines and chemokines. Yes, when you inject, so induce alarm signals, which are cytokines and chemokines, they show in the green and black dots in this cartoon. And those signals will call for the innate lymphocyte to the local injection site. So you can think of those as the first combat unit to arrive to the local field. So one specific type of lymphocyte named the dendritic cells, because they have lots of dendrites on the cell surface, and they can capture the vaccine, move into the lymph node, and present the vaccine to the adaptive lymphocytes. You can think those adapted lymphocytes as a specialized second combat unit. And the second combat unit cells are now be activated, and they are ready to fight the virus. So as Zach told you earlier that vaccines are not real virus and they are weak. So cells from the specialized second combat unit can quickly win the battle and become experienced soldiers. And those experienced soldiers are called the memory lymphocytes. So the memory lymphocytes are then rest in different organs of the human body, including bone marrow, lymph nodes, and other tissues as well. When the real influenza virus attack again, those memory cells from the second combat unit can quickly respond and become active killer cells or active B cells to secrete lots of antibodies. And Zach will explain what exact antibodies will do. So here shows a real virus attraction that you can activate cells and produce antibodies. And Zach will explain what exactly the antibodies will do.
So most of us have heard of antibodies, but we might not know how they work. So they work in different ways to block infection and spread. So normally influenza attaches to the cells to the interaction with a host cell protein. So here's like inside of a human cell, and this would be occurring inside of a human body. And then the virus would attach to the surface protein and then enter the cell and cause infection. What antibodies do the best is they block the interaction with the cell and the surface protein here. So this is showing a virus covered in these red antibodies, and that's why the virus can't bind to the cell and can't enter the cell and cause infection. Another way antibodies could work is as the virus is budding out from the cell that it was created in, antibodies could block the ability of the virus to leave the cell and therefore prevent the spread of the infection. And there's a lot of ways that antibodies can work at different parts of the virus life cycle. And they can also signal to the backside of the molecule to other cells or other molecules of the immune system to help attack virus-infected cells or just the virus itself. This is what I mean by the backside of the molecule over here. So antibodies are one of the most important parts of that second specialized combat unit that Wei Ping was describing. There are also other parts that play crucial roles that she'll describe.
So as we mentioned earlier that the real virus attack also activate killer cells. There are three common ways that the killer cells destroy the virus-infected cells. So as shown in this picture 8, active killer cells can recognize and bind the virus-infected cells. So killer cells can then release a protein called perforin to form pores on the membrane for target cells, which allow the passive diffusion of granzyme B protein into the cells. And this granzyme B protein will kill the virus-infected cells. Picture B shows you that killer cells can also bind a specific receptor on the target cells called VARs to kill the target cells. In addition to these direct killing methods, picture C shows you that killer cells can also induce indirect killing by secrete cytokines that work from a distance. So TNF-alpha secretion in this case can induce the target cell death as well. So by using all these three methods, killer cells eventually destroy the virus-infected cells. So two well-studied type of killer cells are natural killer cells, we call them NK cells, and cytotoxic CD8 T cells. So as you imagine that larger number of cells die during the virus infection. So after the battle, the field is left with dead virus and dead cells. And those dead cells can leak their contents and the virus to the tissue and cause the tissue damage. We don't want that to happen. So prevent this to happen, the scavenger cells, like a macrophage in this case, quickly come to the field to do a cleanup job. And they are bigger, they are known as bigger eaters. So they eat up the antibody-bound virus, and antibody-bound virus infect cells. Therefore, the scavenger cells play a critical role in limiting lung injury and inflammation caused by the influenza virus infection. This is just a simple model. In real setting, there are many, many more parts of the immune system in the human body responding to the vaccination. But as an overview, this is how vaccine works to protect you and all of you from influenza infection. However, the immune system does not always work as we want it to be, and Zach will tell you more about that.
As Wei Ping said, influenza vaccines are not perfect in providing protection. This graph shows the variability of vaccine effectiveness of influenza vaccines since 2009. The higher up you go, the more reduction in outpatient medical visits for influenza, or the more effective the vaccine was for that year. So the best year on this graph was 2011, and the worst year for vaccine effectiveness was 2015. So there's also variability within each year. So year to year, there's different vaccine effectiveness, but within each vaccine, there's multiple strains. So sometimes you can have a good vaccine effectiveness against, let's say, the flu A strain that's in the vaccine, but not against the flu B strain. And you might have good effectiveness against people that are of a certain age, let's say over 65, but not as effective with people under the age of 5 or something like that. So there's a lot of variability even within a single year. And there's two main reasons for this. One is the virus, and one is the person who gets vaccinated, or the host. And I'll discuss a little bit about the virus before Wei Ping will go on to discuss the host. So the main reason why it doesn't always work is because the virus keeps changing. And that's the same reason you need a new influenza vaccine every year. So in blue is an influenza virus that comes from people. It has 8 genetic segments that are in blue. And as the virus transmits from person to person, it mutates, as shown here by these little differences here. And changes emerge over time, creating related but slightly different strains. And if the strain ends up different enough from the vaccine strain, then the vaccine might not be effective. Another way influenza virus changes even more dramatically over time is by something called reassortment. You could think of it as a mix and match genome of the genome segments. So I'm showing you the blue human virus, the black bird virus, and then the red pig virus. If they all three infect the same either person or pig, they can mix and match their genetic segments, and you might end up with a totally new virus. And this is typically how pandemics get started, creating a new virus quickly. It's a powerful diversifying force. And, one virus can outcompete all the others because it just happened to have a good collection of different virus genes. And so the virus can mutate, it can reassort, leading to changes in the virus, which can lead to failures in the vaccine that was, produced in February for the October, November, December season. And for influenza vaccines to work more often, they need to be able to deal with this universe of influenza viral diversity that's out there. The goal of the influenza vaccines of the future is to create a universal influenza vaccine that covers all of them. And so let me explain that just a little bit further. So you can have a vaccine against a single circulating strain. This one is the A Hong Kong 2671 from 2019. That's what's in the current vaccine this year that you're getting or have already gotten. And it's represented by one virus over there. So currently we have 3 or 4 distinct strains like these into the vaccines. So it covers, different HA types and different strains of flu, but it's just individual strains. You could further encompass more related strains if you could cover all H3 and 2 strains, for instance, or multiple HA groups, or all HA groups for all influenza A, or you can cover all A and B strains. So right now we're just taking select viruses from different groups of viruses, but we're nowhere near covering all of these types of viruses in a single flu vaccine. So to make a truly universal flu vaccine, though, you probably want to have it cover multiple years. And so that's where hopefully we'll be heading in the future. That's the goal. But even if you could cover all this viral diversity out there, which currently we cannot, there's still another issue impacting flu vaccine effectiveness, which is the person that you vaccinate, and Wei Ping will explain.
Good point, Zach. So studying both human and animals suggests that several host factors can impact influenza vaccine efficacy. So immunohistory is one of them, which acquire over the time through both virus exposure and vaccination, which can affect the quality and quantity of antibodies developed against influenza virus later in your life. So the human body is also inhabited by a diverse microbial community called a commensal microbiota, which can impact the flu vaccine efficacy as well. Other studies show that the obese adults immunized with the flu vaccine are at 2 times greater risk of developing influenza or influenza-like illness. And pregnancy is another factor that also associated with the physiological and immunological changes which can impact vaccine-induced immunity in humans and cause the vaccine failure. So biological sex and gender are social factors are another impact factor. Research shows that in young adults, immunization with a seasonal vaccine results in at least 2 times greater antibody response in female than males. Last but not least, age also plays a role. So in general, the clinical vaccine efficacy estimate in older adults are lower than in young adults. So to explain all these factors that impact vaccine efficacy, let's take age as an example. So the immune system becomes less able to cope with infectious disease while aging. As shown in this picture, showing that many arms of the immune system, as we referred before as both the first and the second combat unit, soldiers in the combat unit, in the old adults are less responsive to the vaccination. So the specialized dendritic cells we mentioned before, which can act like a messenger to bring the vaccine to the second combat unit, such as the killer cells, which destroy the dead cells, and also the B cells, which, very important part, produce the antibodies. All these cells are less responsive to vaccination in old adults. That is why vaccine does not work well in old adults. There are some scientific data showing the antibody response to seasonal flu vaccine are reduced in old adults, and here is some data. So in this study, the blood samples from young and elderly were tested for antibody response after flu vaccination. We look at the HI titers, basic titer towards the hemagglutinin and protein. So the fold increase of antibodies induced by flu vaccine was significantly higher in young adults as compared to the old adults for each of the flu strain included in the flu vaccine, including influenza A, H1N1, H3N2, and the influenza B virus. So to overcome this, there are some strategies available to increase the immunogenicity of a flu vaccine in people with a weaker immune response. Slide will show you some examples to improve the flu vaccines. Fluzone high dose is the only licensed high dose flu vaccine, which contains 4 times the amount of antigen vaccine. So these high dose vaccines are intended to give older adults a better immune response and therefore protect them from infection. 8 is a standard dose flu vaccine contains an adjuvant, which is approved for use among people 65 and older. So the purpose of adjuvant here is help promote a better immune response, so can protect the following infection. Fluzone intradermal and the flu vaccine skin patch are alternative route of flu vaccine administration. Intradermal injection uses a tiny that on vaccine patch consists of 100 solid water-soluble needles that are just long enough to penetrate the skin. More approaches available as well, the virus-like particles, as Zach mentioned before, a hollow core virus particle, which express the virus protein on the surface to mimic the real virus infection. Also have another approach using the viral vector to express flu protein. So the virus vector the flu vaccine does not rely on egg to produce the vaccine, and also enable delivery of the vaccine towards the mucosal, since the virus vector itself tends to go to the mucosal site. We have other forms of DNA and RNA vaccine can be produced in the lab, which are less expensive and faster than the traditional vaccine production. So these are all some examples. Teams of scientists from all over the world are working really hard to develop better vaccine to protect us from infection.
In summary, flu vaccine causes immunity by essentially imitating a natural infection by various formulations to induce immunity, which consists of experienced primed immune system, including memory cells. However, unfortunately, the vaccine is not 100% effective and does not always work due to viral changes over time and viral diversity, as well as the challenge of some vaccinated individuals to develop protective immunity. Nonetheless, vaccines are constantly improving, and we are working towards the goal of universal vaccines to cover these problems. With that, thanks for listening, and we'll be happy to take any questions. Thank you very much.
I don't know if it's science or just something I've read, that if you do the thing called intermittent fasting, that it kills off your old cells and you activate your T cells to grow a healthier immune system in an older person. Is that a thing, or is that just diet talk? That's all I got.
I don't know too much about intermittent fasting and vaccine responses personally, but I could imagine it could have an effect.
I have a question. I'm not sure whether you're ready for me or not. I'm wondering whether there's a bunch of formulations for COVID-19 vaccines that are using the mRNA platform, including the Pfizer and the Moderna vaccines. And one of the great things about them is this kind of rapid turnaround where, given the genome of the virus, mRNA vaccine can be put together in a matter of weeks. And the cost of fabrication is pretty low and it's pretty fast too. Is there currently an emphasis on looking for mRNA vaccine platforms for flu so that the lag time between identifying strains and coming up with a deliverable vaccine could be reduced to much less than several months?
The answer is yes. So as Zach and I talked in your slides, that there are platforms for mRNA vaccine for flu. I'm not sure whether they're on the market, but definitely research is going on. And I read lots of publications that they are pretty effective.
Yeah, the other part to that is, with flu vaccines, unlike COVID-19, we have stuff that we know works already. So the energy to, improve that is different.
In regards to pandemics, what is it about certain diseases that I guess make it affect so many people? Not obviously we're talking about COVID-19 now, but I guess the comparable example would be, I guess, the flu of 1918. I guess what is it about some of these certain diseases that just cause such an immense toll? Whereas maybe H1N1 certainly caused a lot of impact, but maybe not comparable to COVID-19 and flu of 1918. And especially with, I guess the flu of 1918 wasn't even a new disease. It was also a flu, and the flu's been around for such a long time. So I guess what is it about certain diseases that just wreak so much havoc while others not so much?
There's a lot of study about that, so it's not something that can be completely easily understood. It's a combination of the virus and the host. The virus has to be very transmissible. The host has to be more or less naive to the infection. I think, you're right, flu is around, but because of the changes of the flu, the virus reassorts, you can get something that looks quite new to your body, which was in the case in the flu pandemics, what's happening. And also with the COVID-19, with the coronavirus, we've had other coronaviruses, but this is a new type of one that's more transmissible than perhaps other deadly coronaviruses were. So it is a complex interplay between the virus and the host, and that's all I got on that one. Maybe you have anything to add there?
Yeah, in addition to what Zach said is perfectly right. So there's a combination of virus versus host. So both parties have to make it happen. So virus keep changing and changing to some point that virus maybe bind more efficiently towards the host cells. Maybe host cells have the receptor that really perfect for them, the binding. And all these mechanisms is not really clear, but however, the transmission study we use normally in the lab, we either use fever as a perfect model for the flu transmission that do shows different virus really have different rate, different capability to transmit from one host to another host by different way, either by direct contact or by aerosol transmission. So lots of study need to be done to understand really why this happens. The answer is that it really happened by using the animal model as well.
All right, we've got some questions in the chat, so I'll read those out from Randy. We've got, how do adjuvants help improve an immune response to the flu vaccine? Okay,
I can answer this, Zach. So the adjuvant has different kind of adjuvant. The one licensed now is MA59, which is all emulsion. Basically, they wrap around the vaccine, so bring the vaccine more easily towards the human body, and also inside the human body allow the vaccine to slowly release to the human body to cause a better immune response. And also the vaccine, this is just one type of adjuvant that works licensed by FDA. However, in the lab setting, there's a multiple different kind of adjuvant available. So one of them that our lab working on that is called protein. Protein itself is a combination of those we call it ligand, receptor ligand. So human body has lots of receptor expressed, and those ligands can really engage our receptor and trigger the immune response towards response rate. So this ligand with the vaccine can basically give us stimulation to the human body, and human body responds not only to the vaccine as a foreigner, also respond to this ligand and make the total response better. So the bottom line, the adjuvant is try to help you enhance the response towards the vaccine itself. It's a supplement, it's an add-on to boost the immune response.
All right, so from Christopher in the chat, we got a question about in vaccine development, how is it determined in advance which approach is more safe and effective between mRNA versus other types of vaccines?
I don't know safety studies, and you escalate the dose and make sure that it's handled well by the different participants in the study. You have different phases of the study. That would be, phase 1, then you move to phase 2, and so on. And you compare the reactogenicity and the side effects that happen. If the side effects are tolerable, which has been, published for the COVID mRNA vaccine, and it looks safe enough, then you move on to more and more people. And right now, for the COVID vaccines, we're in phase 3, where they're giving it to tens of thousands of people and making sure that it's safe.
Yes, I try to add on a little bit on Zach's answer. So same as the drug delivery, drug discovery. So the vaccine from lab to the bench to the hospital really takes a long time, maybe less than the drug, but it takes a long time. So always start from design. You need to know all the molecular background to design this mRNA vaccine. Then we always test on the animal model, like mouse and the ferret model, make sure it works perfectly fine. We test, compare with the current available vaccine. Then the next one, move to human study. As Zach mentioned, that you have phase 1, phase 2, phase 3. So all these study, or purpose of this study, try to make sure, number one, the vaccine is safe enough to use on the human. Number two, it's better than the current vaccine before we promote to the commercial. So it's a long way. It takes effort from lots of different fields to work towards that.
Right, and the final thing is the effectiveness. You have to test people who get the vaccine versus who don't, and who gets sick or who gets infected or not, and that's how you tell the effectiveness part once you determine it's safe.
Talking about the effective study, we do lots of effective study for monitor after you get the vaccination, and we monitor vaccine effectiveness on different population, different people, different sites. So lots of work has been put into to really characterize whether the vaccine is effective.
All right, next from Brian, we have a question, are there other common viruses that change often enough to require updated vaccines?
Yeah, I'll say a little bit about this. So there's some viruses that change so much that we can't even get the first vaccine to work. So that's HIV, for instance, is one of those. Hepatitis C is another quite variable virus. I mean, if we did it for coronaviruses over time, we would need to do updates on those as well. I can't think of anything else right now. Wei Ping, do you have any?
Overall, virus change more often than bacteria. So most of the bacteria vaccine seems to work pretty fine, but virus things change too much. Scientists have a strategy to attack that. So normally we try to target on the very conserved region of the virus. So virus, they change, but they not change the whole genome, right? They probably change very often. So that's another approach to get a better vaccine is we try to target the more conserved the part that they change the less. Hopefully they change the less, so whenever they change a little bit on the other part, we still can catch them, still can protect from the infection. So that's another strategy approach that for the flu vaccine.
Great. So the next question in the chat is from Christopher, and he's asking, does the long history of being immunized yearly help you defend from novel viruses?
Yeah, so we definitely recommend annual vaccination. The reason why it is safe before that virus change, and the vaccine you get last year might not protect this year. And also antibody response takes 2 or 3 weeks to happen in your body, but antibody will not last forever. It might decline over 6 months, after 6 months. That's why we recommend annual vaccination. However, there are studies suggest that you continue repeat vaccination or you vaccinate every year maybe affect the following vaccination, but that all depends the strain circulation, what do you get for the vaccine. And I don't think there's a conclusive data now to say yes or no, but there are lots of studies going on. And also, nevertheless, that vaccination it might not 100% protect you from get sick, but even when you get sick, your symptom could be much, much better, could be much, much lighter. So that's why I still recommend annual vaccination.
Another question in the chat from Nadia. We explored monoclonal antibodies as a preventative measure or treatment for flu.
Yes, that has been explored, and there's over the past, I guess, maybe 10 years, there's been an explosion of discovery of monoclonal antibodies against influenza virus. Typically, if you just have one very effective antibody, it might not be as effective as a vaccine, which produces your own natural antibodies within a person. But there are different ways in which monoclonal antibodies can be used, including people are thinking about using them to supplement, let's say, drug-resistant flu viruses. So the answer is, yeah, that is being explored.
I want to add a little bit on that. So that's a very fascinating field that research are going on for that, and use all the new technology, we can sequence what kind of antibody in your body when you're after vaccination or after infection. And we can characterize each antibody and try to test which antibody is more effective. So I'm pretty sure, I'm very positive down the road, antibody treatment will be available, and it's behind the fact that how we want to choose an antibody which is effective, which can last longer, and which can protect you from different strain of virus infection.
Next question in the chat we've got from Rob. The question is, how can a vaccine cause health issues besides allergies?
Yeah, there are some side effects, and depends on the vaccine, depends on the person. Depends if you're using a live attenuated one or not. Your immune system can lead to an autoimmune response. Typically, those vaccines aren't given because they're found to be unsafe. But yeah, if your body sort of reacts to the vaccine, but then perhaps also reacts to other parts of your body that's going maybe overactive, you can have other health problems in it. That oftentimes it's a genetic basis for that. That's triggered by the vaccine, or would have been triggered by an infection if it wasn't the vaccine. Hopefully that's extremely rare nowadays, that we know a lot more than we did years ago. Wei Ping, you have anything for that?
Yes, Zach, I can add a couple of points. So yes, the allergy is probably mostly because the vaccine has the egg component, egg protein, that's caused the allergy. And for that, we now have for this year, we have two different egg-free vaccine, which is cell-based, Zach mentioned, flu cell-based, and also recombinant vaccine. And those people can use the cell-based vaccine or recombinant vaccine. Also, there are reports that flu vaccine causes one of the side effects called GBS, which is a paralyzing illness, which has been reported. But that case was very, very rare. So people should not worry too much about the side effect of the flu vaccine.
Thank you. Next question we have in chat from Brian is there an international standard or body for coordinating vaccine development in the case of pandemics?
So my understanding is the WHO, the World Health Organization, is part of that. Wei Ping, do you know anything more about that?
Not very sure about detail which organization involved, but definitely as the COVID-19 pandemic, when the pandemic happens, every part of the team play a role try to develop the most effective diagnostic as well as vaccine towards the public.
From Priya, we've got, why does our vaccine conferred immunity to some viruses last for years, while immunity to others only lasts a few months?
This is a good one. Well, one reason is how our immune system revs up in response to the vaccines. And if there's not enough, we know a lot from immunology studies in mice and in people, that if you have, let's say, not enough of a reaction to a vaccine, or not enough, let's say, vaccine to begin with, your body sort of starts to rev up, and then as the antigen or the vaccine components go away, your body no longer sees it as an issue, and so it doesn't rev up to create this long-lasting, robust immunity. However, if you have antigen that sticks around for a long time, like in a natural infection, sometimes that will lead to a stronger immune reaction. But there's other things involved, including, the type of virus that you're reacting to. It might not cause a massive response because of the shape of the virus, or because of the other danger signals, like sort of related to what Wei Ping was talking about, the adjuvants that help produce a robust immune response. Wei Ping, you got anything for that?
Yeah, as Zach mentioned, there's many parts play a role why the flu antibody will not last long. Like some antibody towards yellow fever vaccine can last forever. So they're still puzzling us why it doesn't last long, and we are trying very hard to answer that question. But in the meanwhile, lots of approaches have to work towards one of the goals is long-lasting antibody. So the adjuvants, viral vector delivery, all those approaches, one of the goals is try to test. We always test how long the antibody can last in the animal model, in the human body. And I think scientists are aware that that's one of the goals. Not only we want universal, we want long-lasting antibody. You get one shot, you can be rest for a couple of years, don't get any shot. That is important.
I see we missed a question from Rob saying in the case of COVID-19 for vulnerable populations, will it be testing for immunity after the vaccine? My assumption is, although I have no firsthand knowledge, is that, because we do that for flu every year, we're constantly testing vaccine effectiveness in different populations. So my assumption is, that will be followed up on. Vaccine. So Christopher was asking, as a follow-up, vaccine development is just a try and see approach. In some ways, yeah. I mean, but it's based on, centuries of knowledge. So I wouldn't say it's exactly a try and see. And, it's also been refined by rational vaccine design and sort of, predictions. And, but yeah, I mean, you could think of it as, even no matter how much you know and how much you predict, you actually have to try and see.
Yeah, I think science in general is a methodic try and see approach.
But try and see, we come with a very solid background knowledge of what is going on in the body as well as the virus. So we base on the best judgment and to propose the approach.
One last question from Brandon in the chat. I read that dengue fever is unique in terms of the body's response to infection prior to receiving the vaccine. Could you explain why, when it comes to that disease, the vaccine could actually be harmful if used to treat a reinfected person with dengue fever?
So this is antibody-dependent enhancement. Yeah, it seems to be, as far as my understanding, pretty sure that it happens in dengue. There's different types of dengue infections, and depending on which type you get, it can lead to, when you get a reinfection, it being worse because the antibodies don't bind to the important part of the virus that they need to. They bind to a slightly off portion, which somehow the virus uses to attach to cells and enter into cells. So that's a very rare case. That was something people were thinking about for COVID-19 vaccines. Could this possibly be happening with this coronavirus? Doesn't seem to be the case, but it is something that people think about. Certain antibodies could actually help an infection happen. Like I said, it's rare.
Yes, as I cannot emphasize more enough that the immune system is a fine-tuned, highly regular system. So doesn't mean everything is good, everything's bad. It's all about balance. So if you have good balance, maybe towards protection. If you have a bad balance, it's more towards damage.
I don't see any more questions in the chat. Thank you again, everyone, for joining. Huge thank you to Dr. Cao and Dr. Indy. Everyone, don't forget, get your flu vaccine, and I'll see you all at our next virtual science event.
Thanks, everybody.